So, hello everyone. We are once again in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. Today I have with me Yiming Meng. She's a PhD student in the University of Tartu. Under her supervisors are Guillermo Bueno, John Davidson, and Maria Opic in the great group in Tartu University. And Jimin will be talking about with us about a really nice recent paper in Ecology Letters. This paper basically asks a question regarding if phylogeny and environment, which are the effects of these two huge, importantly studied factors in determining plant mycorrhizal traits, specifically status and can you remind me the second one?
Uh, mycorrhizal status and type.
And type. Yeah. So
Yeah.
these are two very important plant mycorrhizal traits that we all want to know about. So, hello, Jimin. Thank you for being with us. And please explain to us how did the study came about? What was the main driving force behind this study?
Yes. Hi, César. Hi, everybody. Thanks for giving me this chance to be here. I'm really excited. Well, maybe I can share my screen.
Mhm.
I hope you can see some slides here.
Yes.
Well, this study was inspired mainly from this paper of Plano in 2017. If you look at this figure on the left side, we see that plant mycorrhizal types didn't show a very clear pattern along latitude or elevation, but mycorrhizal status showed some patterns like increase or decrease. And on the right side, there's also this map about mycorrhizal distribution in Europe. And if we check the last two panels, we see that OM, obligatory mycorrhizal plants, are more common in the south, while facultatively mycorrhizal plants tend to be more at higher latitudes. So what's really interesting or fascinating sorry, about this study is how it underscores the importance of compiling data on plant distribution and mycorrhizal status on a large scale. This is especially true for plant mycorrhizal status because it hadn't been extensively studied before. So our aim is to expand this kind of analysis to a global scale and do this with greater detail.
Yeah, that's great. That's great because I knew about this literature from Guille's work for many years, and it was always in my mind exactly what you just mentioned. How can we expand this kind of analysis? Because of course, for Europe, you have very well detailed plant data in relatively small grids, but how to expand this to the whole world. So I see the inspiration. You you just mentioned two plant mycorrhizal traits. Are there more plant mycorrhizal traits that you think that are important?
Yes, well, the common or the main plant mycorrhizal traits we often talk about are plant mycorrhizal type and plant mycorrhizal status. So plant mycorrhizal type looks at morphology or evolutionary aspects of the symbiotic relationships. This includes the categories like avascular mycorrhizal, AM, ectomycorrhizal, EM or ECM, and also ericoid and orchid mycorrhizal. And if you think some plants never form symbiotic relationships, so these plants can be non-mycorrhizal and M types. And there's also a trait called mycorrhizal status.
Mm.
It describes whether a plant always form mycorrhizal associations. So this is obligatory mycorrhizal, or some plants form mycorrhizal association under some conditions. So sometimes being mycorrhizal, so these are facultatively mycorrhizal species. And again, for those plants never form mycorrhiza, they're non-mycorrhizal status. So non-mycorrhizal NM can be a status 2. So these are the two traits we often talk about, but there's some more traits. One is called mycorrhizal flexibility. So it describes the ability of a plant to grow with or without mycorrhiza. And there is also mycorrhizal dependency. It describes how a plant benefits from or relies on these fungal associations. Like we can measure biomass of a plant, like maybe with mycorrhiza, the biomass can be higher, and without mycorrhiza, then this biomass can be super low. Then we say, okay, this plant has very strong mycorrhizal dependency. And these traits are actually very well described, in this paper from Marimora in 2014. So these are the 4 traits in her paper. But I would say it's important to note that the definition of mycorrhizal traits isn't set in stone. Like it's not like has to be like this. It really depends on the specific research question, how you classify these relationships, or even personal preference in scientific interpretation. Like this paper, this very nice review from Bala last year, she came up with a framework to describe mycorrhizal traits. Like she actually came up with 3 different aspects to describe to describe mycorrhizal traits, like from the aspect of symbiotic mycorrhizal traits, or plant mycorrhizal traits, or fungal mycorrhizal traits. And imagine the trait about plant mycorrhizal type we discussed before, like AM type or ectomycorrhizal type. In her paper, they're actually put in the symbiotic mycorrhizal traits. So I definitely recommend checking this paper for some fresh perspectives.
Yeah, that's great. And I think that those symbiotic mycorrhizal traits, I would call them also like emergent traits, because are traits that just emerge from the presence of both the fungi and the plant. So these are traits that cannot be like classically assigned to an isolated individual, but rather arise from the interaction, which is of course a main focus that we want to study. Can you dip a
Yeah.
little bit further in the facultative status, because this is always that something calls my attention. And there is a recent review from Jan Janssen, Thomas Kuiper, I think, on AMF, and maybe they say that it's not so clear when a plan is facultative. I am thinking that because maybe sometimes you, for example, look for colonization, but or for arbuscules, if we are talking about AMF. But of course, this is a dynamic process, and is a process that changes through the year.
Yeah.
How much there is root colonization. So, can you dip a little bit further in how exactly to determine when a plan is facultative?
Yeah, it's always kind of controversial about facultatively mycorrhizal species, but for us, my short answer is we do some detective work in literature, which means we focus on publications. publications in peer-reviewed literature with empirical original data, meaning that they really went to the field to look for the species or doing basic experiments in the greenhouse. And we paid much attention to the methods. We look at whether this research is based on morphological or molecular analysis. For those studies conducted in the field, our focus shifts to microscopic slides and the statistical tables. These help us determine the mycorrhizal type, because if there are some slides, we can view the structures of maybe AM or ECM. So this help us determine mycorrhizal type of a plant. And also we can find if these individuals, because they are always replicates, if every individual of plant species is colonized by mycorrhiza. Let's say if a paper, they go to the forest looking for a pine tree species or spruce tree species, and it has 10 replicates, and each individual got colonized by ectomycorrhiza, then we say, okay, this is ectomycorrhizal species and also obligately mycorrhizal species because all these individuals were colonized.
Mhm.
So in the case of greenhouse studies, well, we always have notes about each literature we checked, like if this is in the field or if this is in the greenhouse. Then if it's in the greenhouse, then it's the inoculation process. We compare the control groups that are not inoculated with those are inoculated with like AM spores. So the comparison is quite important for understanding if a plant can grow with or without inoculation, which also help us define facultatively mycorrhizal plants. And this is somehow our standard.
Oh, that was a great explanation. Now going into the introduction of your article in Ecology Letters, you explain that some approach to determine at least the mycorrhizal type is to extrapolate to whole plant genera or families, which this is a quite famous paper from 2018. So what do you think are the advantages or disadvantages to do that, to do this extrapolation?
Yeah, I can have a very long answer for this question.
Yes.
So, well, actually, after this famous 2018 paper published, we also have some like other discussions, or also some other papers were published in 2019 about this global distribution of plant mycorrhizal traits. If you look at the one on the left side, it's a Nature paper, and the one from Kamal, it's a Nature Ecology Evolution paper. Both are very great papers. So the paper on the left side, it studied 28 plants, 28,000 species. And the paper from Kamal, she studied 200,000 species, which is super impressive.
Wow. Yeah.
And these two papers, well, they both describe global distribution of plant mycorrhizal type or plant mycorrhizal status, covered a lot of species. So these two papers are based on extrapolations, which means if we use this method, like we say maybe plant species within the same order or same family or same genus, they may have similar mycorrhizal trait. So this is like extrapolation. With this method, the advantage is quite obvious that we can describe many plant species from like this 200,000 species. So there are also some disadvantages, I would say, like we need to bear in mind that extrapolating traits based on their relatives may overlook some details, like how mycorrhizal symbiosis influence plant adaptation, or speciation.
Mm.
And within a single genus, it happens that even within the same genus, different species may have different mycorrhizal types or status. So if we do this extrapolation based on family or based on genus, we may say, okay, this whole genus is AM, but actually there are some actual mycorrhizal species here or non-mycorrhizal species here. So we can make mistakes. So this is the advantage versus the disadvantages. It always depends on your question. If you want to like describe more species, of course, extrapolation can be very useful, but maybe it can be not that precise.
That was a great answer. And also there are a lot of dual plants, right?
Yes,
A lot of
exactly.
dual species. So it's not possible simply to extrapolate at least
Yeah.
to those dual. So this brings me to the a question that I always ask in this interview, which were the main questions of this article? Which was the principal question in this article?
And so if we take into our previous discussion that using empirical data or using taxonomic data, or by using taxonomic, I mean extrapolation. So if we think about these two very great papers, they are based on more or less extrapolation. We think, okay, we still lack such studies based on real empirical data. So this is our first knowledge gap. Maybe we can use the real data or the data without extrapolation. And also, we were thinking that maybe using taxonomy or phylogeny is not enough to describe global distribution of plant mycorrhizal traits because plants live or they stay in the soil and their growth can be hugely affected by the climate or the soil. This process may also shape this symbiosis. So we also hope to take environmental conditions into consideration. So our first question is to check phylogenetic consistency. This question is particularly relevant in our earlier discussion about this, the role of phylogeny. So we hope to know how consistent are plant mycorrhizal traits across different plant lineages. And our second question is more like descriptive, like what is the global distribution of patterns of these plant mycorrhizal traits? And the third question is the role of environment. So what is the separate role of phylogeny and separate role of environment in shaping plant mycorrhizal traits? And what is the combination of these two factors in shaping plant mycorrhizal traits? And also, since we consider environmental drivers of plant mycorrhizal traits, there are actually some previous very nice studies already showing us, like this paper from Barcelo proved that environment, this is like the first principal component of environmental factors. It actually explained 44% of AM species abundance. But in contrast, soil variables, here we have bulk density, explained only 2% of AM abundant. So from this paper, we see that, okay, for AM species, for actual mycorrhizal species, environmental factors can be a very important driver, but soils are not that important. That's why the title is called Climate Drives. But in the same year, I think, not in the same year, 2 years later, we know he had another study in the Pyrenees. In his study, he also showed that like mean annual temperature can be quite important variables for plant mycorrhizal types. But soil variables like pH, like cation exchange capacity, and nitrogen can also play a role. So there's something similar between these two studies that, okay, environmental variables are important for plant mycorrhizal traits, but also some difference. One realized that, okay, the soil doesn't matter, but another said soil is important. So this is also something I'm quite curious about. What is the role of soil? So, well, we are studying environmental variables, so we hope to include both climatic and soil variables to do some comparison to see, okay, which one is more important? Are soil variables really not important, or we like neglect or we miss something? Because one possible reason is that in this Journal of Ecology paper, Barcelo, they worked with 100-kilometer scale. And in this mycorrhizal paper, Bueno, they worked with 1 kilometer scale. So maybe the scale of the study also matters. That's why we have these 4 questions to do some description, to study the role of phylogeny, the role of environment, and to really pick out those environmental drivers that influence plant mycorrhizal traits.
I always have that discussion with Guille regarding the scale, because when you go into a forest, you see the variation. I have some plots here where, for example, plant available phosphorus changes from 3 parts per million to 35 parts per million in 100 meters. You see a huge variation. And of course, we all know that plant available phosphorus is very important for AMF at least. So that brings me to the methodological question. Of course, I always encourage this, the objective of these interviews is for our listeners to go and read the paper is what you should do. But can you explain us a little bit about the methodology regarding which plant phylogeny did you use? Which was the scale that you just mentioned about the scaling for soil data? Which were the soil data? Where did you obtain the plant mycorrhizal traits themselves?
Yeah, as for scale, in this paper, we use 1 kilometer, which
Correct.
is a relatively fine scale, I would
Yeah,
say, because if
yeah,
we think of previous studies, maybe they're at 50 kilometer or
yeah.
100 kilometers. So we're really going to a smaller scale at 1 kilometer.
Mm.
And we have mostly four data sets. The first one is the plant mycorrhizal trait data, which is this empirical data we were talking about. We really looking into published literatures for each species and list their, like, okay, how many papers support this AM and maybe how many papers support this non-mycorrhizal status. We have a table of this,
Mm.
and you can actually find this table in the supplementary of this paper.
Yeah,
So this is our first database. The second one is plant occurrences data, because we know, okay, this species is AM, this species is orchid mycorrhizal. We hope to know where are they. That's why we use GBIF, a global biodiversity information facility, because here we have these occurrences worldwide. We got their coordinates. So this is our plant occurrences data. And because we hope to know the role of environment, we have these climatic and soil environmental data. These are basically raster layers, also at 1 kilometer scale, but they're from different sources or databases like CHELSA or Soil Grid, and also some other published results. Then you need some, of course, cleaning of this environmental data, because maybe the 1 kilometer grids are not exactly overlapped. There may be some cross there. You have to like resample all this environmental data to make sure that they're in a perfect stack. And our last data set is this phylogenetic information. There's a R package called v.phylomaker. So this package can provide you a megatree, and this megatree was based on a paper from 2018, and this paper includes phylogenetic information of 75,000 plant species. So within this megatree, we can pick out which species we want, and these are species we have plant mycorrhizal trait information. So we basically work on these 4 datasets, which you can all find in our supplementary or our open data in Dryad.
Mhm.
And doing data cleaning or filtering can be a little bit painful. I can keep talking about this for the whole day because I really spend a lot of time cleaning the data. But the short version is those coordinates from GBIF, some are maybe not reliable because maybe people put in the wrong coordinates or different projections. So you have to be very careful about this data.
Thanks. So can you tell us one thing which is always important for me or and for many of us regarding the percentage of plant mycorrhizal types? Because we always cite Smith and Reed or the 2018 paper we mentioned before, but which is the updated number with your paper?
Well, I can't give the like this is saying that this is the right number or
Yeah.
correct number
No, I know.
because
I know.
here, yeah, in our paper, I mean, the database we have, plant mycorrhizal trait, I mean, empirical mycorrhizal trait data is around 14,000 species, which is in the supplementary.
Mhm.
But as you know, I did some data filtering, maybe ruling out those species with wrong coordinates or very like local or endangered species because they're not that representative. And after doing this data filtering, I have 11,000 species in my data set. And as for my result, we have like 72% of AM and around 11% of ectomycorrhizal species. For orchid and ericoid species, there are about 1%. And as for mycorrhizal status, I would also say that obligately mycorrhizal species are dominant, about 68 or 70%. And facultatively mycorrhizal species are around 18%, and non-mycorrhizal species are around 14%. So this is in our data. If you go to some other database like the famous one, Fungal Root, I guess they provide more information about plant mycorrhizal types and status. The number can be different a little bit, but I think mostly the trends should be the same.
So interesting. Can you tell us, because we have like 5 minutes, maybe the main result, how important were both phylogeny and environment for status and type?
Yeah, well, I would say that plant mycorrhizal traits are closely related to both phylogeny and environment. So for mycorrhizal type, they're more, if we check this table here, we did variance partitioning. So for mycorrhizal type, we say that phylogeny is more important because it explained more than 40% of plant mycorrhizal traits, and environmental variables explained only 7%. But well, this is also what has been proved preferred. Basically, we have similar results align with previous studies. But what is neglected or has not been very well studied is plant mycorrhizal status. We see that for status, environment and phylogeny are basically similarly important, explained for around 14% of plant mycorrhizal traits, meaning that if you study mycorrhizal status without taking environment conditions into considerations, you may miss a lot.
Mm. That's a significantly more, a significantly higher percentage that I imagine. So, and proportionally is about a third, less than a third or more than a third of how important is type for type. So that's quite interesting. And what about? Uh, I'm interested, sorry, in the plant families which have both types, maybe, and the distribution across families of plants of type and status. I guess that
Yeah.
Ericaceae and Eucalyptus, which were the results regarding plant families?
Well, maybe you can check this phylogenetic tree for more information because we listed the 15 most abundant families in our database.
Mhm.
And something need to pay attention. Maybe this pine family, as you see, everything has been like estimated as ectomycorrhizal, but actually there are many dural species here, both AM and ectomycorrhizal type. The reason why we put this dural mycorrhizal type as ecto is because their niche in the forest or in the outside are quite similar, and it's better to put them together for like better data availability, like for more observations. But in general, for most plant families, I won't say they have only one type or only one status. Like even for those like orchids family or Ericaceae family, we still see that some species are not ericoid mycorrhiza or not orchid mycorrhiza. There are always some non-mycorrhizal species here. And also for like Raniculaceae, this family, it is reported that this family is highly dependent on avascular mycorrhiza. But if we check very carefully, we still see something pink. It should be non-mycorrhizal species here. So it's always like you can't say the whole family has only one type or only one status. It always depends.
So interesting. Sorry to rush, but can you also show us the maps? Because
Yeah.
your maps do reflect the original predictions from David Reed, or they are not so similar? What do you found?
Well, I think for the map of avascular mycorrhizal, this is not very surprising because we know that avascular mycorrhizal species are more abundant in the tropical area or in the warm area. So we
Yeah.
found a lot of distribution here. But one thing is that we have, I guess, because these occurrences are from GBIF, we have a lot of records in Europe, in North America, but we lack, or not lack, but we have relatively less observations from South America, from Asia. That's why maybe this pattern showing here for South America is not precise enough because compared to those occurrences in Europe, in North America, we have really like limited occurrences here. And for actomycorrhizal, it basically peak in cold and acid region in northern hemisphere. For ericoid mycorrhiza, again, we found a lot of ericoid mycorrhizal species in cold and acid soil in North Europe. And there should be also a lot of ericoid species in the Andes. But again, the data is kind of limited in South America. So I really hope to get more information about this part. And non-mycorrhizal species, again, here we found a lot in Australia, because if you imagine this very harsh environment in central Australia, this is also what we were expecting. The trend for facultatively mycorrhizal and obligatively mycorrhizal are basically contrast. Like we found more facultatively mycorrhizal species in the higher latitude, but less obligatively mycorrhizal species there. They peak more in the tropical and warm regions.
So many thanks for that. At the beginning or at the middle, I guess you mentioned that one of the main questions was regarding, I think the fourth question of your paper, it was regarding the environmental drivers of both types, of both traits, sorry. So can you tell us about that?
Yes, we have a bar plot in Figure 4 introducing these important environmental variables for mycorrhizal type and status, and also for each mycorrhizal trait. And we can check these last two figures that I would say for mycorrhizal type and status, the important variables are not important. Sorry, are not same. They're different. Like on the left side, we found that soil texture, like sand, and variability in soil properties like pH, are key predictors for plant mycorrhizal type. And climatic variability, like standard deviation of this isosomality, which is, can't find it right now, but like for, so like soil textures, again, the standard deviation of clay, and also the mean of clay. And so these variables like were quite important for plant mycorrhizal types. But for mycorrhizal status, climatic variables, especially solar radiation and evapotranspiration, are like most strong predictors for plant mycorrhizal status. And variability in temperature, this is standard deviation of temperature seasonality, and also wind speed were also quite influential. Also, soil factors like histosols and soil textures and also pH can also be predictors of mycorrhizal status. And if we look deeper into different mycorrhizal types and status, they have its own unique set of environmental drivers. So I can't say that there's a general trend for all the traits, but I would say each mycorrhizal trait got its own set of environmental predictors.
So interesting the wind speed, for example. I never imagined so, but yeah, it's conceivable that I mean, it's very probable that it has a huge effect. And also, so your last point was so interesting that each, for example, each mycorrhizal type has its own drivers. uh, One of my last questions regards the main conclusion. I think that you maybe already mentioned this when you show us the table one, but still, can you tell us what was the take-home message from this study?
Yeah, so wrap everything up, I would say we hope to explore how plant mycorrhizal traits are distributed and what factors influences them, whether it's environment or phylogeny. Our aim or goal was to understand the balance between how these traits are evolved in response to plant phylogeny and environmental variables. And we found that there's a significant variation in how strongly these traits are conserved across different plant species. We found that mycorrhizal status tend to change, or in the paper we see more liable compared to mycorrhizal type. And both these two traits are influenced by a variety of climatic and edaphic, meaning soil factors, and soil textures, as we see in this bar plot, maybe can be a particularly important factor.
Great. Which do you think that are the next steps for this? Because something that caught my attention was the 11,000 species that you mentioned, but this is still, I mean, I realized that this was a huge amount of work. All the data that you created, all the GBI presence
Yes,
data
indeed.
coordinates, it seems like it was years of work, but it still is like 4% or 5% of all
Yes.
plants that
I
we
would say
can.
maybe less than 4% of vascular plants.
Wow, wow. So what are the next steps?
Well, I'm not sure about if I really have a very clear picture of what will be the right thing to do. But what I'm doing now is, as you've seen in the picture, in the paper, I'm mostly doing this description work to describe its distribution worldwide, to describe to doing this variance partitioning. So now what I'm doing now is do some prediction because we have these environmental variables already. We have this phylogenetic information already. So we're trying to think that if we can predict plant mycorrhizal trait using different predictors, if we can use only environmental variables to predict plant mycorrhizal traits, or using the combination of environmental variables and plant phylogeny together to do this prediction, to
No.
see like which combinations of predictors can better predict plant mycorrhizal traits. As you see, we have only 4% of data available.
Yeah.
It helps us to at least predict something we're not know for now.
Mm.
And with some prediction, maybe we can, I don't know, in the future, maybe when we have more empirical data, we can do this validation to say, okay, if our model or our prediction is good enough.
Oh, that's fantastic. That's always the next step in macroecology, I guess. So,
Yeah.
and this is also a next step for you in your career, or are you about to finish your PhD? What are your plans?
Well, I'm doing this prediction right now, so I hope I can finish this. Well, I'm in the middle of data analysis, but I have some results already. So I hope I can have this prediction paper published as soon as possible. Then I can do my defense. So this predictory part is basically part of my PhD. After that, I don't have a clear picture for now. It's like I always have no plan, but I don't know.
Oh, okay, okay. Well, thank you very much. It was
Thank you.
an
Thank you.
illuminating discussion. Is always a interesting topic. I'm always thinking about. I have published like this basic data. Like here in Chile, we publish something of 255 species, I think, and it's just the mycorrhizal type in a book chapter. And I always want people to publish this kind of information. It seems very basic to some, but it's so important to publish this somewhere.
Yes, yes.
So I encourage our listeners to do so. And thank you very much for the paper and for your time. And I
Thank
hope
you. Thank
to
you.
see you at ICON maybe.
Yes,
Okay.
in August.
In August. Okay, okay, that's
Yeah.
great. Thank you so much. Bye.
Thank you. Thank you.